Nucleic acids are present in the nucleus. The nucleic acids are combined with protein called nucleoprotein. Nucleoprotein containing protein molecules and nuclein. The nuclein is also called nucleoprotein with nucleic acids. Nucleic acids combined with protein called nucleoprotein. In this nuclein or nucleoprotein, protein is eliminated. Remaining parts are called nucleic acids. The nucleic acids are also called nucleotides. Nucleotides are mainly containing two major components. They are phosphoric acid and nucleoside. Phosphoric acid and nucleosides are present in the formation of nucleic acid. Then nucleoside again containing two major components. One is carbohydrate and second one is nitrogenous bases. Carbohydrates are also called sugars. They are again divided into two types. One is deoxyribosugar, second one is ribosugar. And the nitrogenous bases are two types again. One is purine, pyrimidine. Purines are two. They are adenine and guanine. Pyrimidines are three. They are cytosine, thymine, and uracil. It is the total composition of nucleic acid flowchart. And based on these nucleic acid, nucleotides are containing phosphoric acids and nucleosides. The nucleosides are again two. One is sugar molecules and nitrogenous bases. See children, every DNA or every nucleic acid containing phosphoric acid as a backbone. The phosphoric acid chemical formula is H3PO4. H3PO4 is a chemical formula of phosphoric acid. Each phosphoric acid is connected with two sugar molecules. Each phosphoric acid connected with two sugar molecules with diester bonds. Between one phosphoric acid, one sugar molecule, one ester bond is there. One phosphoric acid connected with two sugar molecules with two ester bonds. So we called phosphodiester bonds. It is the ester bond. Phosphodiester bond. Phosphoric acid act as a backbone of nucleic acid or nucleotides. And comes to the sugar molecules. The sugar molecules are two types. One is deoxyribosugar and ribosugar. See children, it is the deoxyribosugar. Deoxyribosugar. On the second carbon of ribosugar, deoxyribosugar, one oxygen atom is eliminated. One oxygen atom is eliminated. So oxygen atom is eliminated on the second carbon called deoxyribosugar. Deoxyribosugar. So if oxygen is present on the second carbon of sugar molecule called ribosugar. See here it is, oxygen is absent on the second carbon of sugar molecule. Each sugar molecule shows pentanone ring-like structure. Pentanone ring-like structure, and it contains 5 carbon atoms. First carbon atom, second carbon atom, third carbon atom, fourth carbon atom, fifth carbon atom. Fifth carbon atoms are here. The chemical formula of sugar molecule CHO. CHO means carbons, hydrogens, oxygens are present. See, here it is. Carbons are 5. Oxygens are 5. If oxygens are 5, we called ribose sugar. If oxygens are only 4, we called deoxyribose sugar. And H10, C5, O4, H10 is a chemical composition of deoxyribose sugar. C5, H5, sorry, C5, O5, H10 is the ribose sugar chemical composition. Each pentanone sugar is connected with nitrogenous bases. With nitrogenous bases, glycosidic bond. Between the nitrogenous bases and sugar molecules, glycosidic bonds are present. These are the glycosidic bonds. See children, here it is, the glycosidic bonds are again divided into two types. One is C1N9. C1 means it is the sugar molecule carbon 1, first carbon is connected with ninth nitrogen of nitrogenous bases. Ninth nitrogen of nitrogenous bases. If the sugar molecule is connected with purines with the glycosidic bond, the glycosidic bond is called C1N9 glycosidic bond. C1N9 glycosidic bond. If the sugar molecules are connected with the pyrimidines with glycosidic bond, that glycosidic bond is called C1N1. C1N1 glycosidic bond. See on this diagram, it is the deoxyribose sugar. It connected with the purines of adenine with glycosidic bond. With the glycosidic bond name clearly we called C1N9 glycosidic bond. So glycosidic bonds are again divided into two types based on the purines and the pyrimidine structure. There are two types. What are the two types? C1N9 glycosidic bond. C1N9 glycosidic bond means C1 it is the carbon of sugar, first carbon of sugar. N9 means it is the ninth nitrogen of purines, ninth nitrogen of purines with glycosidic bond. If the purines are replaced with pyrimidines here in this diagram, here it is C1N1. C1N1 glycosidic bonds are present. So it is the glycosidic bond. And we comes to the nitrogenous bases. The nitrogenous bases are two types. They are purines and pyrimidines. Purines are double ring structures. Purines are in double ring structure and pyrimidines are in single ring structure. First we discussed about the purines. Purines are two. One is adenine and second one is guanine. See children, on these purines, two rings are present. One is benzene ring and second one is imidazole ring. Second one is imidazole ring. On this benzene, first carbon, third carbons are replaced with nitrogen atom. See here it is, carbon atoms are absent. The carbon atoms are replaced with nitrogen atom. So first carbon is replaced with the first nitrogen, third carbon is replaced with third nitrogen. It is in benzene ring. The benzene ring it attached with the one is pentanone ring. The pentanone ring is called imidazole ring. On this imidazole ring, seventh carbon and ninth carbons also. Seventh carbon and ninth carbons also replaced with nitrogen atoms. First carbon, third carbon, seventh carbon and ninth carbon. These are replaced with the nitrogen atoms. So these are the double ring structures. These are connected with sugar molecules with glycosidic bond. The glycosidic bond is C1N9 glycosidic bond. If comes to the pyrimidines, the pyrimidines are single ring structures. See, these are the pyrimidines. The pyrimidines are single ring structures. In this pyrimidine, cytosine, thymine, uracil are here. In benzene ring, first carbon, it is the first carbon and is the third carbon. The first carbon and third carbons, these two carbons are replaced with the nitrogen atoms. If these pyrimidines are connected with the sugar molecules with glycosidic bond, that glycosidic bond is called C1N9, C1N1, C1N1 glycosidic bond. N1 is a pyrimidine. C1 is a carbon atom of sugar molecules, but N1 is a nitrogen, first nitrogen of pyrimidines. So between these two nitrogenous bases, again hydrogen bonds are here. The hydrogen bonds are two types. One is double hydrogen bonds and triple hydrogen bonds. Always adenine bind with thymine. Always adenine bind with thymine with two hydrogen bonds. And guanine always bind with three hydrogen bonds. Guanine always bind with cytosine with three hydrogen bonds. It is in the DNA formation. If you take RNA, the thymine is replaced with uracil. The thymine replaced with uracil. So we observed these three types of chemicals. What are the three types of chemical? One is phosphoric acid, one is sugar molecule, one is nitrogenous bases. Each phosphoric acid connected with the two sugar molecules. Here it is one phosphoric acid, it connected with the two sugar molecules with two ester bonds. It is one ester bond, it is another ester bond called phosphodiester bond. Phosphodiester bond. And the sugar molecules are connected with the nitrogenous bases with glycosidic bonds. It is the glycosidic bond. The glycosidic bonds are again divided into two types. One is C1N9 glycosidic bond, C1N1 glycosidic bond. C1N9 means if the sugar molecules connected with the purines, sugar molecules connected with the purines, the glycosidic bond is called C1N9 glycosidic bond. If the sugar molecules connected with, if the sugar molecules connected with pyrimidines, sugar molecules connected with the pyrimidines called the glycosidic bond called C1N1 glycosidic bond, C1N1 glycosidic bond. And between the two nitrogenous bases, hydrogen bonds are present. Always adenine bind with thymine with two hydrogen bonds. Always guanine bind with cytosine with three hydrogen bonds. So during the formation of nucleic acids, these are the three chemical bonds. One is phosphodiester bonds, and second one is glycosidic bonds. Third one is hydrogen bonds. It is in the formation of DNA. If we comes to the RNA, in RNA thymine is absent. The thymine is replaced with the uracil pyrimidine. Uracil pyrimidine. So it is the chemical composition of nucleic acids. Chemical composition of nucleic acids. See children, it is important for essay question.